Welcome everybody to Science and Wine Club. Glad to see some familiar faces here, some fellow board members and some new faces as well. And so I'm excited to get our event started tonight. I'd like to introduce myself. My name is Cindy Haba. I am a PNRI board trustee and chair of our development committee, and I have the honor of hosting and emceeing tonight's event. So I know we've got some folks that are still joining and coming on board, so we'll continue to let them in as they're joining the show. And but we'll just get started with some let you know what we're here to do tonight and how this evening is going to go. Science and Wine Club. We know that science has become a primary topic of discussion these days. I think everybody is interested in learning and reading and hearing about science in all different ways. And here at PNRI, we want to encourage and to fuel that interest and to be a part of and to be a leader of those types of conversations. And as many of you probably know, PNRI has a long history of pairing the stories of science with wine through our many events over the last 15 plus years. And so just about a year ago, we came up with this idea of Science and Wine Club as a way to engage our audience. If you remember, about a year ago, we were kind of still in our virtual world, and the explosive interest in science overall that was happening. And we wanted to find a way to bring together our scientists, our staff, our board members, and most importantly, our donors and our audience, such as you all, to share in the same passion for learning about cutting edge science. So that's what we do here. We share our learning about science, and we get to drink some fun wines as well. And I'm so happy to say that this is our fourth Science and Wine Club event this year. And so welcome to those of you that are new. Welcome back to those of you that have been here before. And I would love it. Go ahead and post in the chat. Tell me how many Science and Wine Club events you've attended. 1, 2, 3, 4. Let us know kind of who's in our community here with us tonight. We've had some pretty amazing events, the last 3 session events that we've had. In the very beginning, we had Dr. Rick McLaughlin when he talked about the evolution of our genes. And our second event was Dr. Lisa Stubbs, where she talked about stress and genetics and her new research project on that topic. And then we had Dr. Michael Metzger give us an update on his cancer research, where he uses clams to understand why some cancers are resistant and others are not. And all of those prior sessions are available on our YouTube channel if you go to our PNR website to check those out if you missed any of our previous sessions. And so tonight we're going to be hearing from Dr. Bill Hagopian and talking about his research in type 1 diabetes. And I also would like to mention, before we get too far into this, our sponsor, and to thank Collins Communications and Brenda Collins, who is with us here tonight, for sponsoring our event. Thank you, Brenda. So at PNRI, we are making a conscious effort to put science first, which is why this is called our Science and Wine Club. And our researchers are at the center of everything that we do. And we feel it's important to hear straight from them and straight from the source, which is why we love bringing their voices to you directly. And so we have our science and we have our wine. But because we're going to start with Dr. Hagopian first, does not mean that you can't jump in and enjoy your wines now. We do have Big John from Sheridan Winery here with us tonight to talk about his wines. And so we will get to his part of the agenda a little bit later, but go ahead and open your wines now. Enjoy them while you listen to Dr. Hagopian give his talk. We're going to throw in some tasting notes in the chat. So if you have the opportunity to have some of Sheridan wines, you can read a little bit about them before Big John talks about them. If you have anything else you're enjoying, don't wait. Go ahead and make yourself comfortable. Thanks for showing off our wines that we have. Thank you, Sherry. Thank you, Brenda. So that we can engage in our conversation here. And so I think I'd like to introduce Bill now. And I've known Bill for a long time, and he's one of the main reasons that I've been affiliated and partnered with PNRI. And I could probably gush about him just generally about his contagious enthusiasm for his work, his determined and endless energy. But I actually have a formal introduction that I'd like to share so that we all can share and know about his expertise. So Dr. Bill Hagopian joined PNRI in 1997, and he leads our diabetes research program. He's a practicing physician and a clinical professor at the University of Washington Medical Center, where he's treated patients with type 1 diabetes for over 30 years. Bill is an esteemed leader in the international diabetes community. He served on numerous boards and committees, including the Immunology of Diabetes Society, the National Institute of Diabetes and Digestive and Kidney Diseases, the Scientific Advisory Committee of the University of Washington's Clinical Research Center, and the TEDdy Consortium. He's published over 150 manuscripts and reviews and was recently published in the esteemed Nature magazine. His research focuses on bench to bedside strategies in immunology and molecular biology to predict, prevent, and to treat type 1 diabetes. And so without much further ado, Bill, I'd love you to come on board and say hello and take us through all of your information you'll share tonight.
Thank you, Cindy, for the very kind introduction. And I really love being at PNRI. It's a great home in which to do this research. And only 30 years is not enough to solve a disease like type 1 diabetes, but we're making some progress. Let me share my slides. And let's see here. There we go. Let's
Great.
see that.
We can see that. Thanks, Bill.
Wonderful. Okay. My talk is going to be about neutralizing childhood type 1 diabetes and celiac disease. And later in, during the Q&A, I can talk a little bit more about the more comprehensive approach to all of type 1 diabetes, including people that already have it. But right now I'm trying to talk about early prediction and prevention. So let me see if I can change my slide here. There we go. So let's see here. I got to get my Zoom. Got to get my Zoom control out of the way. So type 1 and celiac disease are autoimmune diseases, and that's where the body gets confused and attacks self. And the vicious cycle over here on the upper right are the cartoon drawings of the cells in the immune system that actually are doing the damage. This yellow cell is an antigen presenting cell, which picks up antigens from, for example, a dying beta cell, and then chews them up and presents them in this molecule called HLA and presents them to a helper T cell, which then stimulates a killer T cell, which goes and kills the antigen, which is found on a beta cell. And this goes around and around and around and around. A bystander effect is that this helper T cell also helps B lymphocytes, which then make autoantibodies, which circulate in the bloodstream. All this other stuff, which is kind of the important stuff, happens in the pancreas. And we can't sample it in people that are living, and not until they die. And so we have to actually use these antibodies to try to predict what's going to happen, what's happening with this other branch. And this cell could be an insulin cell, or in the case of celiac disease, it could be an intestinal lining cell. So the autoimmunity is the same sort of process that goes on. Down at the bottom are some sections, histological sections, which show cells, and each cell nucleus is a little dot here. So you can see this is actually a part of the pancreas, and this round thing here is an islet of Langerhans, and it's a blob of about 1,000 cells, and most of them are insulin secreting beta cells, but it's being invaded by these little black dots. And the little black dots in the lower left-hand picture are lymphocytes, which are invading and doing the things that are happening in the cartoon up above in real life. On the lower right-hand side is the intestine, and up at the top are little finger-like projections called intestinal villi, and they are part of how you absorb things in your intestines, absorb nutrients, and they get attacked in celiac disease. And as you can see in the bottom, there are lots of those little black dots in between the villi, and the villi are all broken down and short. So this is intestinal atrophy that occurs in celiac disease. Now, I should say that these are not rare diseases. Type 1 diabetes occurs in 1 in 300 kids, and celiac disease, 1 in 300, and celiac in 1 in 100. So these are fairly common diseases as they occur. Let's see here. Okay, And it's not really normal, you wouldn't think, for the immune system to be attacking self. And so what starts this out? Well, TEDdy, the whole idea of TEDdy, the environmental determinants of diabetes in the young, it was to try to find what the triggers were. And one of the major places that we looked was in the intestine for microbes, microbes being bacteria and viruses. And it turns out that they're prime candidates, and we found some, and here we got on the cover of Nature magazine, which is pretty cool. There's your teddy bear. So we've made a splash in the research community, and that's great. Okay, so what might be triggering type 1 diabetes? Well, here we go. Enterovirus can be seen living in the insulin cells. And these are two sections from two different patients. The lower three are one patient, the upper three are one patient. The upper three are from a patient in the JDRF NPOD study. The lower ones are from a Scottish histologist that collected samples from kids that died of type 1 diabetes 30 or 40 years ago. And what these show on the left in green is that there are insulin producing cells in the pancreas, and these are serial sections next to each other. So they show kind of the same islet, just a different slice right next to the other one of the same islet. And here they're staining for the enterovirus shell protein. So this shows that the virus is actually in these cells. And if you merge the two, you can see that actually that the viruses are in the same cells that stain green. So they're in the beta cells. And my interpretation of this is over here. It's a bullfight. And the bull is the immune system, and it finds it very hard to ignore these viruses living in these beta cells. And that may be what triggers type 1 diabetes, at least in some of the kids that get it, is that the virus is waving a red cape at the bull, and the bull is charging and attacking. And it starts by attacking the virus, but it ends up by attacking proteins that are specific to the beta cell. So this is an interesting story, which has a lot of supportive evidence. It's not proven yet. It has a lot of supportive evidence, and I think it's an intriguing story. It's a little bit different in celiac disease, and in celiac disease, the triggering antigen is gluten that's present in wheat, and gluten has a protein called gliadin. That's the irritating protein that gets presented to the immune system and triggers celiac disease. But how does that gliadin actually get into those to get to those cells in between inside of the villi? And that's probably intestinal infections. This is also a theory, so I'm warning you that this is just what I think probably happens. And so if you get an intestinal infection and you have a lot of gluten in the inside of your intestine, and you have the genetic predisposition to celiac disease, then it triggers celiac disease. So this is a graph from the TEDdy study published in 2019 in the journal Gut. And as we go up, we go from low to middle to high gluten consumption. And as we go rightward, we show that an enterovirus infection has a greater risk of triggering celiac disease in those with higher intakes of gluten. So in this case, the infection and the gluten trigger work together. And the interesting thing is, for celiac disease, it appears that a lot of different intestinal infections can work with gluten to trigger this. Even viruses that don't really cause a lot of disease, like reovirus, have been published to trigger this. And rotavirus, which is now prevented by a vaccine, but also enterovirus, our friend from the previous slide. But enterovirus may be a little more specific in type 1 diabetes and triggering. Here, it's just any intestinal infection. This is what I think that the situation is. But again, some of it's my opinion and not completely proven yet. Okay, here is two graphs. The graph on the left is about type 1 diabetes. On the right, it's about celiac disease. Okay, so let's start with the one on the left. This is age from starting from birth all the way to age 12. And the graph shows when people develop islet autoantibodies during their infancy and young childhood. This is data from TEDdy, where we were sampling every 3 months, starting at 3 months of age. And as you can see, the islet autoantibodies really start appearing at about age 9 months or 1 year of age. And there's a big peak in the first 4 years of life. And then there's another smaller peak that occurs about from 8 to 12. And so what this suggests is that there may be different triggering events that occur in different kids at different ages. But it's very, very intriguing and suggests that this disease is a disease that gets triggered in very young children a lot of the time. Now, on the right is a graph of appearance of transglutaminase antibodies in celiac disease. And the ages at which these antibodies appear, showing celiac autoimmunity, are also very young. It's a little bit of a different distribution, but it seems that between ages 3, 4, 5, 6 are the peak, and by the time you're 11, most of the children that are destined to get celiac disease have already gotten the disease. Okay, now they may not be diagnosed for another 10 years until they're in their 20s, but they appear to get the disease when they were quite young. And that's my interpretation of this data. Again, not proven, but this is kind of what I think has happened. And it's a shorter period of time for those here between when they got their transglutaminase antibodies to when they got diagnosed with celiac disease, sometimes with an intestinal biopsy. Then over on the left with diabetes, where oftentimes kids that get their antibodies really young don't get their diabetes until 5 or 8 or 10 years later. Okay. Now, why would we want to predict diabetes ahead of time? Well, one of the real problems that occurs is diabetic ketoacidosis, where the bloodstream gets very acidotic, the pH is off, and it's a life-threatening condition. The kids that get this are often lethargic, and they can even lose consciousness. They get admitted to the hospital, often to the intensive care unit. And what this shows, this is data, early data out of New Zealand. And I think it's been replicated more recently with similar data out of Colorado, but I showed the New Zealand data here. And the red is the incidence of type 1 diabetes onset by age. And as you can see, there's kind of a plateau, but it kind of peaks around puberty. And that's kind of a typical pattern that we see everywhere. But the black bars are the proportion of those cases that have diabetic ketoacidosis. And as you can see, among the very young, when they get their diabetes, almost all of them have diabetic ketoacidosis. Between 0 and 1, the few cases that get type 1 diabetes, all of them have ketoacidosis. They're all very, very ill and get admitted to the hospital, usually the intensive care unit. They can have brain swelling. In the old days, they used to die. And that's those pancreas sections I showed you. But now, it's very rare to die of type 1 diabetes, DKA, but sometimes it's touch and go. It's very serious. As they get older, a little bit less often they get ketoacidosis. And then again, there's another peak kind of around the teenage years when kids tend to neglect themselves and not go in until they're about ready to fall over. So this is kind of DKA, and it's a serious life-threatening disease. It's expensive, causes a lot of illness, and it can be prevented. So this is 4 different papers showing, and these were from different studies, the DAISY in Colorado, the TEDDY study, which is 6 centers, Baby Dive in Germany and DIP in Finland. And they all show a background rate of ketoacidosis, DKA, of between 23 and 44%. And they were all dramatically decreased in kids that were followed with autoantibodies. And you can see that the rates go down a lot. The p-values, which is scientific for significance, are very low for these. These are very highly significant events. Any p-value less than 0.05 is considered significant by scientific custom, but these p-values are so low, they're highly, highly significant. So this is an important effect that prediction can have on preventing serious illness in those kids that are getting a new type 1 diabetes. And the way that it's prevented, one of the ways it's prevented is educating parents of children at higher risk. So we make traffic signs like this, which show the early symptoms of type 1 diabetes, and we give them to our study participants and our patients to show what the early symptoms are that they might get and that they should look for in their children that have islet autoantibodies. Sometimes we do other things like monitoring their blood sugars sort of loosely. They don't have diabetes yet. They may not get it for years, but when they do get it, they can get very ill unless it's detected early. This was designed by my clinic manager, Michael Killian, who I think does a great job for the studies. I'm very grateful for his work. Okay, and what's another thing that we can do? Well, we hope we can actually apply immunotherapies to those kids that have autoantibodies but don't yet have diabetes. And this is the first study to actually show that. It's led by Kevin Herold, who is a professor at Yale now, and he's a real leader in this field. It's an anti-CD3 monoclonal antibody. And this was given in TrialNet to kids in families with diabetes that were autoantibody positive and at risk of diabetes. Actually, they were autoantibody positive and had very early signs of abnormal blood sugars, not yet diabetic. And by giving teplizumab, the onset of diabetes could be delayed. These were randomized to one of two groups: the group given salt water, or the group given teplizumab, and the group given teplizumab had a much slower rate of onset of type 1 diabetes. And this isn't a cure for diabetes, but it's very, very promising, very significant difference between the two curves. And it's the first example. So obviously this can be tweaked, it can be adjusted, and it's going to get better and better. And there are also other agents that are very, very promising, anti-CD2, anti-TNF, that actually are very promising to try to do the same sort of thing. So Kevin published this in the New England Journal in 2019, so it's actually quite recent. I should put in a plug for myself here, a shameless plug for myself, that the first study published on teplizumab was by Herold and Hagopian and others in the New England Journal in 2001. So Kevin's been working on this for a long, long time. All right. Next, I just showed you how we might be able to prevent or at least delay type 1 diabetes. And this is a theory about gluten that was shown in data from TEDdy that got published in the Journal of the American Medical Association. So highly important stuff. And this, interestingly, was not an experiment where we randomized people to one treatment or the other, like I showed you on the previous slide. This is just observing what their gluten intake was and how often they got celiac disease. And it shows that those that had a lower gluten intake were less likely to get celiac autoimmunity, in other words, transglutaminase antibodies, and were less likely to progress to celiac disease, which kind of makes sense because it's the environmental trigger. But it sort of highlights a way that we might be able to intervene. And again, this is not proven because nobody's done a randomized controlled trial yet. But this is a great idea that by telling the people that have super high genetic risk for celiac disease, or that have celiac autoantibodies already, to take a diet that's low in gluten, maybe not zero gluten, but maybe 1/3 of the normal amount, will significantly delay or prevent them from going on to get celiac disease. This is a hazard ratio of 1.75, meaning a decrease in disease of about 40%. So this is very promising. And again, significant p-values. Okay, but what is the problem with being able to predict these diseases? Pediatric prediction as envisioned in these typical studies that follow kids, lots of kids, and do very, very frequent antibody testing is just not cost effective. And this was published in Pediatric Diabetes back in 2015 by some of my colleagues from Florida, and they actually suggested that it wasn't cost effective. And we'd known that for a long time. We'd been working on making it cost effective while they published this. So they played right into our hands here by pointing that out. How do we make type 1 diabetes prediction more cost effective? Well, less expensive clinical management, which could be done through a website or app. Less expensive autoantibody follow-up tests, which can be done by better sampling and better lab testing. More specific pre-screening so that less kids have to get the autoantibody test, hopefully without losing a lot of sensitivity. And that is what our genetic risk scores can do now. And then combining to find multiple childhood autoimmune diseases. Okay, so this is the website example. It worked very well. 80% of Do It families did this website, and we can both have them sign up for kits, get contact information, give them results, have them find out about the disease. The next thing is a less expensive autoantibody sampling collection. We have kits that we send out in the mail. They prick the finger, get a few drops of blood into a tube, mail it back to us. Antibodies and DNA are both extremely stable in the mail, and so this works great. More than 70% of our Do It families were able to do this successfully, which is really, really impressive. And so this was able to save a lot of money and a lot of time for the families, but a lot of money for the study as well. So this is a very hopeful thing. And then the next thing I want to show you is maybe some more advanced autoantibody assays. We have for 20 years, we published 20 years ago about radiobinding assays for islet autoantigens like insulin GAD, IA2, and zinc transporter. But now we can make fusion proteins of these islet antigens with deep sea shrimp luciferase, an approach that is championed by my colleague Vito Lampasona in Milan, and also by Peter Burbello at the NIH. And these have shown to be very, very effective. And you could make this protein, stick it in your deep freeze, and then just take out a tube every week and do assays, and it's very effective. So this is hopefully going to make things cheaper than the radiobinding assay tests that we use, and hopefully as accurate. Okay, so the last thing I want to tell you about to save money is the most major thing of all, which is actually doing a genetic risk score screen before doing the islet antibody tests, and just doing the islet antibody tests on those that are at the highest genetic risk. And of course, those that are positive for autoantibodies, we can educate them, give them immunotherapy, monitor their blood sugars, but we can do this in a stepwise fashion to be more efficient. And that's what the Cascade study, our new study, is about in Washington State, called the Combined Antibody Screening for Celiac and Diabetes Evaluation. Okay, and so the genes that we look at, our favorite one is HLA, because it's the most important gene. It's the one that carries the highest risk. And HLA is like a serving tray that a butler might have. And on it, it serves pieces of antigens that then stimulate the immune system. And they may be pieces of a microbe like one of the enteroviruses. They may be pieces of an autoantigen like a piece of glutamic acid decarboxylase, or GAD. And these are served to the immune system to stimulate the immune system. And so HLA types are really important in type 1 diabetes. But there are a lot of other genes as well with lesser importance. This HLA over on the left here is Mount Everest, but these other ones are definitely important foothills. And so we'd like to be able to factor in all of these genes in the risk in determining the genetic risk of type 1 diabetes and of celiac disease in order to predict who needs to have these autoantibody tests to watch them. And with some great colleagues at the University of Exeter, Rich Oram, Mike Whedon, Seth Sharp, and Lorique Ferrat, we developed a genetic risk score. This is I was involved with development of the second generation genetic risk score. And we use a term for determining the quality of this as the area under the curve. And the blue line here is no better than random. This green line is better and clinically useful. A perfect test would be this red line. And they're all described by the area under the curve here. And the red line has an area under the curve of 100%, the green line 80%, and this random chance one is 50%. So this one has a AUC of 0.927, which shows it's very, very good, the genetic risk score GRS2 for type 1 diabetes. We have a similar one for celiac disease. Here are the HLA molecules on the left. Here's the other genetic loci in the middle, and the area under the curve of the raw curve is 0.881. So very good for celiac disease, very good for type 1 diabetes. Okay, how do we use these? Well, we screen the whole population at birth using a leftover spot from the newborn screening program. So the state legislature mandates a newborn screening program. They test for 30 different diseases. When they get done, there's several spots left over. We get permission from the parents to take a punch of one of these spots, and we do an assay for all 70. We do about 70 SNPs. And what this means is that we can calculate a risk score for type 1 diabetes and another risk score for celiac disease. And then we have to choose a cut point above which we would follow these kids. If we choose 85% cut point, that means we're only following 15% of the kids, but they have 93% of the early type 1s and 90% of those diagnosed up to age 5. But if we choose a cut point of 90%, it's cheaper because we have to follow less with the autoantibodies, but we detect in the 80s. So this is a way that we can actually make this cost effective enough that we might be able to get the newborn screening program to take it on for the first couple of years, and payors like United Health Group or Primera Blue Shield or Kaiser Permanente, who are paying for people's medical care to adopt this for regular medical care. Oops. Okay, and once we do that, we can follow up with antibody tests. If I can get the right slide. Ooh, this likes to do 2 at a time. Let's see here. So after maybe more frequent screening at infancy, in those that are at the highest risk, we can then screen only at ages 2 and 6 to follow up after that. This is a combination of studies called TIDI, which was calculated by IBM and sponsored by JDRF. And what this shows is that if you sample autoantibodies at only age 2 and 6, you can catch 82% of all the diabetes that occurs before age 15, between the ages of 2 and 15. So that's pretty good for cost effectiveness. Only 2 tests. You take 15% of kids, you just do 2 tests during their childhood, and you can detect 82% of those that are getting diabetes. The 15%, I think we were at 90%. So 90% times 80%, 72% total of the cases detected. And we've done it on the cheap. And we hope the payers will actually pay for this because it probably saves them money because it's so cost effective. All right. Yes.
I was going to say that we want to save some time for questions at the end, and and give a little bit of time that we get to hear from Big John as well and get to taste our wine. So I want to just encourage folks that if you have questions, you're welcome to pop them into the chat right now, and or you can raise your hand when we get to the Q&A in just a couple more minutes, so that we can you can get a chance to ask Bill anything directly from what he's told us.
Okay, can I do 3 more? So this is the problems with celiac disease, and we can do a similar, very efficient testing strategy for that to save money. And the Cascade study is going to do this. And this is my lab group and all of our sponsors over the years. Awesome. We've been very grateful for all of these sponsors. Thank you.
That's great. Yes, thank you, Bill. So feel free to throw your questions in. We're going to turn this over to Big John right now and share a little bit about the wines that that we have highlighted this evening. Big John, you want to tell us a little bit about them?
I'm on. All right.
You're on.
They had to wake me out of my slumber. It's so much science and a little overwhelming for a guy like me that doesn't have any kids and doesn't have to worry about juvenile diabetes. But thank you, Bill, for all that great information. Many of you are familiar with Sheridan Vineyard. We are an estate winery in Zillah, Washington. Many of our neighbors refer to us as the gorilla in Zillah. That finger usually gets pointed at me, but we have 3 terrific wines that come from the estate. So we have 100 acres. So everything that says Sheridan on the label is from us. We're grape growers more than anything. It's been a difficult and very interesting harvest for all of us. The summertime was extremely hot, and for all of you in Seattle without air conditioning, I was with you. It hit 122 in the vineyard this summer. Blew records away. We did get some damage in the vineyard in the Merlot because we couldn't get enough water to the vine. So, but for 2019, which is the first wine we're going to try tonight, is the Mystique. This is our kind of lower tier Bordeaux blend, Cabernet Sauvignon dominant with a fair amount of Cabernet Franc and a little bit of Merlot. No new oak on this whatsoever. So this goes into a barrel that's been used one time. So you will get a little wood tan into it. But for the most part, this is a super soft and juicy and very fruit forward wine, and something that can go with barbecue. It can go with fine dining. If you've got Wagyu beef, this would hold up very, very well. This is a wine that you can lay down and cellar for a few years, but the pure joy of drinking this, and I'm going to let you know. It is juicy, it is soft, it's got nice richness, it's got some beautiful acidity on the back end. This is such a pleasure. This is what we call the Monday through Wednesday wine here at our house. We usually have a number of bottles open so you can taste them over a couple of days. And the sign of a really great wine is if the next day it's better or as good as the day before, you're on the right track. So if you ever find a wine that kind of falls apart on you the next day, usually it's a science project and it's made in mass. We do about 2,000 cases of Mystique, sometimes 2,500. So this is our second highest production of all the wines that we make. And if I was still a dating man, and my wife loves this story, we've been married 28 and a half years now, Mystique would be my wine of choice to ply my potential mate. So just throwing that out for anyone who is dating these days, or whatever that case may be. On to the next wine. We're going to do the Boss Block. We're going to do the Cab Franc next. This is our second smallest production. We do about 300 cases of this every vintage. It is kind of a cult wine for us. The name Boss Block comes from our owner's wife, Karen Greer. She wanted a wine named after her. And Scott, our owner, said, well, Karen's Cuvée doesn't sound all that great, but how about we name the entire original block planted in 1997 after you, honey, because you're the boss. So we have Block 1 Cabernet that was first, and then Boss Block Cabernet Franc right next to that. This wine is from the original blocks, so planted in 97. It does see about 50% new oak. So this has some definite wood tannin to go along with the old vine skin tannin that you would get off of vines of that age. Cab Franc can have some herbal and green notes to it, and our trick in not capturing that pyrazine or pyrazine kind of note is we keep the crop very low. We stress the vine. It's easy to do in this part of the world because we have, the hot days, the cool nights, we get very little precipitation. So if your vines are not stressed in Eastern Washington, you're obviously watering way too much. And I say watering way too much. During the heat of this year, we watered 24 hours a day, 7 days a week for 5 weeks. We've never had irrigation cycles like that. And so the fruit, for the most part, is in, and the vintage looks good. The crop is much smaller than previous years. We're down about 30% because of all that heat. But that's Mother Nature's way of leaning out the crop. And aromatics are great, colors are great, berries were very, very small. So it's going to be a heck of a time for Cabernet Franc. So let me shut up and have a little of this. That's pure elixir right there. We tend to pair lamb with the Cab Franc a lot, whether it be rack of lamb, lamb, lamb shank stew. My famous lamb burger recipe, which I'll share with you guys at some point. It's an easy one to to fall in love with. And we've made a lot of converts for people that don't care for Cab Franc. I want to throw something out to everyone. There was an opportunity to buy on the Sheridan website, and it was kind of a debacle with the PDF and all that, and we apologize. What I would like to offer to everyone involved here tonight is we will honor those prices through the end of the year, and basically that's membership price. So if you are a member, you get a ridiculous discount on these wines. Our membership is only 10 bottles a year, 6 in the spring, 4 in the fall. You never pay a tasting fee. You get to come to Zilla, to our beautiful garden, and taste with your friends, and it doesn't cost you a dime. If I'm there, I'll feed you because I'm winery chef, and that's always a good gig. But we want to offer you those prices, and call the winery. There'll be a phone number on there, and that's for Jenny Schmidt. And she runs membership, and she's a doll, and I've known her for years. And when we looked at expanding to this new facility, I told Scott that we needed to have Jenny, or I'd pull my hair out, and I don't have a whole lot left to pull. So that was kind of that would have been a deal breaker for us. On to wine number 3. Syrah is our lease production wine in the vineyard. We have 4.5 acres. It's in kind of a draw, so it's in a really warm section. It faces south and southwest. So during all this heat, it really gets baked, and you can taste that in the wine. You know, it's got a savory, really smoky characteristic to it, but at the same time, it's got a blueberry pie sweetness that is really inherent to that section of the vineyard. We only do, maybe 300 cases total of Syrah. We have a upper tier Syrah that we do as well that sells for $90. This one is a relative steal for $20 to $25. So it's a fun one. Syrah is a very chameleon-esque variety grown in Eastern Washington. It can be really gamey and weird, full of pickle brine and funky notes out of Walla Walla, or you can have, high tone, high pitch acid, and with blue and black and red fruits in it that you get from the Yakima Valley. So quick note on the Yakima Valley, almost 40% of wine grapes grown in Washington come from the Yakima Valley. The first American viticultural area ever recognized in the Pacific Northwest was the Yakima Valley. The oldest producing red and white grapevines come from the Yakima Valley. So when you go visit your friends in Woodinville and Walla Walla and, love all those guys, their fruit comes from us. Walla Walla produces less than 5% of all grapes grown in Washington State. The Walla Walla AVA is mostly in Oregon. So they have dual citizenship. I'm an Oregonian, so I'm not going to diss them on that. I love my Walla Walla boys, and they support PNRI just like we do at Sheridan. So,
Hey, Jeff, we're gonna.
Yes.
Yep, we have a lot of questions coming up into the chat room. So we want to give folks a chance, both you and Dr. Copian, to answer some of those. So take a sip and tell us how good the Syrah is, and then we're going to jump to some questions.
Bill, I hope you're getting some of this, because damn, that's good. On to you, sir.
Excellent. So thank you. That was great information. I'm a Mystique fan, so love Sheridan Winery. Thank you, Big John. So Bill, I'm going to jump to a lot of these questions in here, and thank you everybody for participating. There's a lot of interest in what you're doing in your research, and a lot of questions around obviously the connection between celiac disease and type 1, and does having one increase your risk of having the other? And how does gluten impact your risk of developing type 1 if you have celiac? If you already have celiac, do you have a higher chance of getting type 1? So some similar questions around that. Can you just speak a minute about the connection between those two that you know of, that you study?
Yeah, we were lucky because in Teddy we studied both diseases, so we could see those people that intersected and had both, and those that were separate. It turns out that DR3, DQ2.5, is the high risk haplotype for celiac disease, and it's also a high risk haplotype for type 1 diabetes. So there's a lot of automatic genetic overlap between the risk of the two diseases. And there's a slight increase over that in the amount of overlap that you would see, more than what you would expect based on just that, but very, very little. So actually, I would have to say that my speculative point of view is that they're not. That gluten doesn't really trigger type 1 diabetes, and that having celiac disease doesn't necessarily trigger type 1 diabetes. There's a caveat. I mean, obviously, enterovirus is one of the intestinal infections that can increase permeability of the intestinal lining, so the gluten triggers celiac disease more efficiently. So that may be why we saw that little increase. But some studies have shown yes, some studies have shown no. And when you see that over the years, after my first 30 years of research, I just have to say that it means that it's a small effect, if any.
Okay, great. I'm just going to go in order here. And just so you know that if we have more questions, we will be following up and we will be posting answers to all your questions. So if we don't get to it tonight, know that we still will have Dr. Kopian speak to those and get those answers out to you. So we have a question regarding teplizumab. Just curious that if the FDA declined to approve it, do you have any comments on that?
Yeah, so teplizumab was a great and pioneering monoclonal antibody for type 1 diabetes. It turns out that in my experience, I did 5 studies on this with Kevin. Kevin led them all, and he's a great guy to work with and a great scientist. But it's actually true that it seemed like after the first cycle, and the cycle was a little hard to give. Each cycle was like, think of a cycle of chemotherapy. You had to divide the dose into 10 or 14 different fractions and give them each day for 10 or 14 days, because otherwise you would get too much stimulation of the T cells and you might get a little bit of cytokine storm. So we gave them in fractions over a 14-day period. And it worked really well to stop the immune system in its tracks from attacking the islets. And they would be stunned for approximately 2 years, a year and a half or so. And after that, things would resume and the attack would start again. There was a lot of hope that the second cycle that you might give a year later or 6 months later or something like that might continue that process for a longer period of time. But I didn't see that when I looked at it. And so I was a little disappointed with that. Now, that still doesn't mean it's not an extremely useful therapy, because you could give it and then you might be able to give some other agent as a maintenance therapy once you stop the process in its tracks with teplizumab. And that certainly is the case. But it's very difficult to even get an FDA-approved study for one drug, let alone to get 2 different pharmaceutical companies to agree to collaborate to do a study where you do one immunotherapy and then another immunotherapy after that. And so that's logistically just too difficult, kind of to pull off, I think. So we need probably different agents that are able to maybe induce and maintain the process. So that's just my opinion about it. And I'm not sure exactly what the FDA commission thought was a difficult thing with teplizumab. I think 8% of people have to stop it before they get a full cycle because of the cytokine side effects, but that's pretty small. So I think it's mainly that the second round doesn't act sufficiently well.
Okay, thank you. And we have a question here. This might be well, maybe a quick answer on this if possible. And I know this is from one of our recent esteemed college graduates. So she's got a great tough question here. She's talking about the HLA-DQA1s and HLA-DQB1s, talking about the mutations that coupled with the infections that can be triggers to both type 1 diabetes and celiac. The question is, do the type of infections or the specific mutations on the HLA genes determine whether a kid develops type 1D or celiac?
That's a great question. And all these questions are really, really great, really intelligent audience. Thank you. It turns out that, whereas the DR3-DQ2.5 haplotype is a high risk for celiac disease and a moderate risk for type 1 diabetes, DR4 is more of a high risk for type 1 diabetes and celiac disease. So that kind of more than then whatever particular infection might determine whether or not you get type 1 diabetes or celiac disease. That is the underlying genetics. But it is true that variation in HLA is something that's very important to measure. And the infections, like I said, what my best guess is that there are specific infections that can cause type 1 diabetes, whereas in celiac disease, I think it's GI infections that might just make the intestinal lining more permeable so that the gliadin peptides can get in and do their damage, stimulate the immune cells to do their damage. So it's kind of a different thing there.
Yeah, and these questions are around just the genetic link to that, where there's a question about if you have grandparents, or if you have other family members in your genetic line, does that increase your chances of getting celiac?
It absolutely does. I mean, and for a couple of reasons. You're probably in a country where they eat a lot of wheat, which increases your risk, but also you're inheriting genes that are in your family that are high risk for celiac disease. And the one that comes to mind is that DR3-DQ2.5 haplotype. So it's interesting. Not everybody that has that inherits that particular genetic group of genes will get celiac disease. They kind of have to have these three things happen at the same time. They have to have the susceptibility, they have to have the gluten intake at the time that they get the GI infection. So all those things sort of have to happen together to trigger. A lot of people get very, very weak transglutaminase antibodies and then they go away. So they got kind of a pre-celiac disease, but it didn't really catch, which is great news for people that happens to. And 1 in 100 seems like a lot, but the number of people that have DR3-DQ2.5 as a haplotype are probably more on the order of 5 or 8 or 10%. And to have only the 1% get celiac disease means far more people with this risk haplotype don't get disease than do get disease. Same is true for DR4 and type 1 diabetes.
That's great. Such good information. So I know we have Deborah and Angie and a couple other questions here that we will follow up with some follow-up emails to make sure that we get those questions answered for you. And but I wanted to just do a couple things here before we close out, because we do promise to keep this to 1 hour for everybody's time. If this research has intrigued you, please keep in mind that it's made possible by federal grants, but it is also supported and accelerated by individuals and organizations. In particular, experimental and early stage research and shared resources are supported through direct donations. And so if you're interested in learning more about the research of Dr. Kopian or other researchers at PNRI, please go to pnri.org to read more about our different labs. And tonight's event is sponsored by Brenda Collins of Collins Communications. And Brenda's here tonight. She's also a member of our development committee.
Yes, and I've talked to Dr. Hagopian many times, or actually one time, about celiac disease. I think I talked his ear off because our family is affected. My daughter was diagnosed at 6 and she's now 22. And so we've lived with it for a very long time. But I really am so thank you. I'm pleased to hear all of the research that you've been doing and what PNRI does, and we're big supporters. And just keep doing what you're doing. I'm such a big fan. And here's a toast and a toast to the future of PNRI. Thank you.
Thank you.
Thank you, Brenda. Thank you, Collins Communications. So our next club meeting will be next year. So we round out our 4 sessions this year. We're happy to have all the activity that we've had. And our next one, you can mark your calendars for Wednesday, January 22nd, PNRI's
January
genetic
19th.
I'm sorry. Yes, 2022, January 19th, 2022. Sorry about that. PNRI's genetics researcher, Dr. Amy Dudley, will be presenting, and our winery partner that evening is Long Cellars from Woodinville. So that's going to be another very interesting talk. Dr. Dudley is fascinating to listen to. Thank you so much, Bill, Dr. Hagopian, for tonight's talk and sharing your groundbreaking research and again, your efforts towards preventing, predicting, and solving type 1 diabetes and the link to celiac. And we're really hoping that this testing process, your Cascade project, can come to fruition, and that kids will be tested and that a lot of lives will be saved.
Thank you
and
very much.
yes, and John,
it is true that those people that already have diabetes, there was a pig kidney transplanted just in the news today, and actually what that applies to is that there are cells that can be secreting insulin that might be transplantable to type 1 diabetics, and those are the people that don't wear the artificial pancreas, which is the other solution, which is a machine which actually senses the blood sugar and gives insulin. So there's a lot of hope for people that already have diabetes.
Yeah, thank you for that.
If you don't mind, real quick, having known Dr. H and Big John for almost equal parts in my middle-agedness, thank you both for being wonderful stewards of my health. And I think that you are equally important for most people's happiness and lives. And thank you both for being good stewards of type 1 diabetics everywhere with making amazing wine that can be processed well. And for Dr. H for being just absolutely insane as not only a PI and a doctor, a researcher, but as an individual doctor. I don't know where I would be with either of you. More Dr. H, to be fair.
I understand.
I love you.
I understand.
Okay.
Fantastic.
Thank you both. You're incredible.
Yep. And yeah, Big John, thank you for extending Sheridan's wine discount through the end of the year. There's a lot of Christmas gifts that are going to make people happy under their holidays.
Ho, ho.
Yeah, so that's fantastic. We appreciate that so much. I'd like to give a shout out to our TEDdy study members that are on the call tonight. Thank you so much for being here, and thank you for your participation in research. Truly life-saving work. So we appreciate that. And our guests from JDRF, we're so happy to have you part of us and sharing in this conversation with us as well. The more voices that talk about these topics, the better for everybody. And to our many club members, I was reading through the notes or through the chat. We've got first timers, we've got second timers, and we've got third timers, and we got folks that have been here all year long. So thank you very much for being here. We appreciate you. We hope you continue to join us through our next year Science and Wine Club events. Like I said, we will follow up with any other questions we didn't get answered directly with some emails out to you. You can always check our website again for replays of this video, as well as replays of our previous events as well. And so again, it's been a great evening. Enjoy your wines, and thank you, Bill and Big John, for a great evening.
Thank you guys for listening.
Night all. Yes. Thank you for joining us, everyone, and all the support and all the donations. And let's keep this alive and save our kids.
Absolutely. Thank you, everybody.